Hello everyone, welcome again to my YouTube channel. Today's topic we are gonna start is nucleotides. Before starting my topic, I would like to tell you about the structure in which we are gonna study this nucleotide. The nucleotide topic will be completed in 3 videos. The first video will be related to its general structure and then classification, what type of nucleotides exist, and then second video will be of purines and its biosynthesis, its degradation, and disease related to purine metabolism. The third video will be related to pyrimidines, their biosynthesis, their degradation, and the disease related to their metabolism. So let's move towards the first part of nucleotide metabolism, that is the structure of nucleotides. Firstly, the question arises, what are nucleotides? Nucleotides are the smallest units which when get polymerized then forms a nucleic acid. So here we are just gonna discuss about only nucleotides, not nucleic acids. So let's try to understand what are nucleotides and how many types and all the things about nucleotides. So nucleotides comprises of two subunits. The first one is nucleosides, and the second one is phosphoric acid. Now what are nucleosides? When I try to understand what are nucleosides and break nucleosides into different subunits, then I found that nucleosides also comprise of two subunits, in which one is a nitrogenous base and the another one are pentose sugar. Nitrogenous bases are of two types: pyrimidines and purines, and pentose sugars are of two types: ribose sugar and deoxyribose sugar. Now, this is the general plan according to which we will be studying all the topic general structure of nucleotides. Firstly, we are gonna study about the structure of nitrogenous bases, that are purines and pyrimidines, and then we are gonna study pentose sugars and their structure, and then lastly we are gonna study about phosphoric acid, and then after completing the structures of all subunits which form nucleotides, then finally we will move towards nucleotides and their types, all the types of nucleotides, and some important analogs which are used clinically for treatment of many of the diseases. Okay, so let's move towards the first point, which is the nitrogenous base. The first point is nitrogenous bases. What are nitrogenous bases? Nitrogenous bases are aromatic heterocyclic compounds. Aromatic heterocyclic compounds. What is aromatic and what is heterocyclic? Let us try to understand. Aromatic means ring-like structure. Okay, and heterocyclic means a cyclic ring which comprises of more than one type of atoms. So nitrogenous bases are aromatic heterocyclic compounds, and there are two types of nitrogenous bases. Two types. First one we know, pyrimidines, and the second one is purines. Pyrimidines are further, there are three types of pyrimidines. The first one is cytosine, thymine, and uracil. Purines are of two types, means there are only two purines. One is adenine and the second one is guanine. So now we will try to understand the structures of pyrimidines and purines separately. So let's move towards pyrimidines. Pyrimidines. Pyrimidines are single cyclic ringed compound. Single cyclic ring compound which comprises of six member. We can say it's a six-membered ring. Okay, six-membered, and from these six members it has two nitrogen and four carbon. So pyrimidines is a single cyclic ring compound which comprises of six members, two nitrogen and four carbon. And when we draw the structure of a pyrimidine, then we have to do the numbering, and according to that numbering, there are different substitutes attached on that numbered atoms. So the numbering which will take place in the pyrimidine structure is done according to the clockwise direction. Clockwise direction. Okay. So and now firstly we will see the general structure for all three types of pyrimidines, and then the pyrimidines, there are three types of pyrimidines which are because of different variations of substrate and their positions. Okay, so firstly I am drawing the general structure of all three types of pyrimidine. uh, Pyrimidine is a six-membered single cyclic ring which comprises of two nitrogen and four carbon. So see, This is the general structure for all pyrimidines. Now we will do numbering of all these atoms in clockwise direction, and the numbering starts from the nitrogen, which is just downwards. Okay, downwards means like it will not exist anywhere here. The examiner can like make you confuse to just rotate this ring. Maybe he can like put that this nitrogen in upward direction, and when he will rotate this ring, this nitrogen will be somewhat here and this nitrogen will be somewhat here. So the numbering will be starting from the nitrogen which is on the apex. You can like remember this thing. In pyrimidines, the numbering will start from the apex, we can say like not from the sides. Okay, from apex of the ring. So I started numbering from here. This is first carbon. Then we go according to the clockwise direction. Then second carbon, 3, 4, 5, and 6. So this is all about numbering. Now we will see the substrates according to which we classified pyrimidines in three types. So the substrates and their position. So there are three types of pyrimidines. First one is cytosine. First one is cytosine. Second one is uracil. And third one is thymine. So now try to understand the structure of cytosine. Firstly, we will draw the general structure of pyrimidines. So I drew all the general structures of these. So now, firstly, look forward for cytosine. Cytosine contains one amino group on the fourth number carbon and oxy group on the second number carbon. Okay. Then uracil contains an oxy group on the second number carbon and one more oxy group at the place of amino group. So uracil contains two oxy group, one at the second number carbon and one at the fourth number carbon. And if we talk about thymine also contains two oxy groups. One is on second number carbon and then another is on fourth number carbon. But the difference is what? It contains an additional methyl group at fifth number carbon. So just please balance yourself all the bonds. Okay, so as carbon contains 4 bonds, so we will remove this bond from here. Okay, that's it. So uh, uh, this is the way how you can remember. Let me try to make you understand easily how can you like learn all these structures. Okay, firstly, the main trick to learn the pyrimidine structure, firstly remember in this sequence. If you are trying to draw the structure of this, firstly you will draw the general structure of all the pyrimidines, then you will name them in this sequence: cytosine, uracil and thymine. First one is C U T. You will cut the pyrimidines. Okay, so now for cytosine, you can you can see on second number carbon every pyrimidine is having a oxy group. Okay, so it doesn't matter for the oxy group of second number carbon, it's common in all, but the difference is what? If your pyrimidine is having, if your ring is having amino group, then it is cytosine. If your ring is having oxygen, only oxy group, then it is uracil. And if your thymine is having methyl group also, then it is a thymine. The another way, if amino group present, then cytosine. If not present, then it can be uracil or thymine. And now from uracil and thymine, if it's only oxygen present, then it is uracil. If with oxygens methyl group is also present, then it is thymine. And one thing more, thymine is also known as 5-methyl uracil. Thymine is also known as 5-methyl uracil, because if start to numbering the ring 1 2 3 4 5, on the 5 number carbon it's a methyl group. If I try to hide this methyl group, can you see the same similarities between these two rings? Yes, this is exactly the uracil. So that's why it is also known as 5-methyl uracil. So this was all about pyrimidines. Okay, so now let's start to do purines. Purines. Purines are double ring compound which comprises of 9 members. Okay, 9 members. From these, 4 will be nitrogen and 5 will be carbon. Okay, so double ring compound, and when we will do the numbering of purines, the numbering will be done in anti-clockwise direction. Anti-clockwise direction. So there are two purines, as we know, adenine and guanine. Firstly, we will look at the general structure of purines. The general structure of purines. Firstly, see, as purines are double ring compound, like it will consist of two rings. One will be a 6-membered ring, okay, and one will be a 5-membered ring. Don't confuse that. Oh, 6-member and 5-member, 6 plus 5 is equal to 11, but here are 9-membered ring. Don't be confused. Let's see first. So 6-membered ring is known as pyrimidine ring. This one is known as pyrimidine ring. Okay, because this ring is very similar to pyrimidines, and the 5-membered ring is known as imidazole ring. Imidazole ring. These are the names of this ring. Okay, So let's try to draw these rings. One is 6-membered and one is 5-membered. So this is a 6-membered ring, and this is a 5-membered ring. See, purines comprise of two rings, double ring compound, which contains 9 members. 9 members are 1, 2, 3, 4, and 5. 5 carbon and 1, 2, 3, 4. 4 nitrogen. Okay. So let me balance their valencies. So this is the structure. Okay. Now how will we do the numbering of this structure? We will not start the numbering from the nitrogen on the apex, but we will start from the nitrogen which is on the upper side or on the sides of the ring. Okay, not on apex. So the numbering starts from this nitrogen and it goes in anti-clock direction. So second, third, fourth. Now we will not move towards this ring. Firstly, we will complete numbering of this ring, 4, then 5, then 6. Then after completing the numbering of this ring, we will move from this nitrogen 7, 8, and 9. Okay. So this is all about the numbering. Now according to different substitutes on different position, the purines are also of two types. First one is adenine and the second one is guanine. First one is adenine and the second one is guanine. Okay, so to remember their structure, firstly we will draw the general structure of purines. So this is the general structure of purines. Now let's talk about adenine. Adenine contains one amino group at the 6th number carbon. Okay, but guanine, in case of guanine, we will transfer this amino group from this position to the 2nd number carbon. The amino group is replaced here and the position of amino group is taken by the oxy group. So the same pattern we will try to remember the adenine and guanine. If on the apex position the group is having amino, then it's adenine. If amino is not, then guanine. Okay, so this will be the pattern to remember adenine and guanine structure. If there is amino on the amino group on the apex of the ring, then it is adenine. If amino group is not on the apex of the ring and it is on the anywhere, it doesn't matter, only on the apex of ring, if amino is absent, then it is a guanine. So this is all about purines and pyrimidines. There are some unusual nitrogenous bases which are present in our DNA and RNA. The main function of these unusual nitrogenous bases which are present in our DNA and RNA is to recognize some of the very important enzymes. So some minor or unusual nitrogenous bases present in DNA and RNA. Their main function is to recognize some specific enzymes, and these minor bases are 5-methyl cytosine, N6-methyl adenine, and the third one is pseudo uracil. Some other unusual nitrogen bases are also here, but the most important are these three. Please remember these three. These play a very important role. Okay, so now the second point is pentose sugars. Pentose sugars, there are two types of pentose sugars, D-ribose, and the second one is D2-deoxyribose. Okay, D means dextrorotatory. It's all about concept of chemistry, organic chemistry. So don't worry what is this D means. So the D just normally means dextrorotatory. Okay, it's some phenomena for this light. Okay, so now let's try to draw their structure. 5-carbon sugar, okay. The difference between D-ribose and D2-deoxyribose is ribose sugar is having hydroxy group on the second number carbon, and the deoxyribose is having only hydrogen. The oxygen group is absent in deoxyribose, okay, at the second number carbon. Let me do the numbering of these carbons. So 5. So this is the D2-deoxyribose, means the ribose sugar in which on the second position oxygen group is absent. So this is D2-deoxyribose. D means dextrorotatory. Okay. So this was all about pentose sugar, and now the term comes is phosphoric acid. Third one is phosphoric acid. Chemical formula of phosphoric acid is H3PO4, and if we try to draw the structure of phosphoric acid is somewhat like this: P double bond O, OH, OH, then OH. This is phosphoric acid, and to form nucleosides, it is attached to the pentose sugar in the form of PO4 3 minus, which can be drawn like this. Okay, so this was all about phosphoric acid, pentose sugar, and nitrogenous base. So now we have completed all the structures of all the subunits, which finally results in the formation of nucleotides. Now we will discuss about nucleotides. There are two types of nucleotides. First one is ribonucleotides, and the second one is deoxyribonucleotides. Ribonucleotides, when polymerized, they result in the formations of RNA, and deoxyribonucleotides results in the formation of DNA. Okay. Difference between ribonucleotides and deoxyribonucleotides. There are two differences. First one is the sugar which is included in the ribonucleotides and deoxyribonucleotides, and also nitrogenous base. One nitrogenous base is different in both. Here uracil is absent, and on the place of uracil here is thymine present. Okay, but here uracil present but thymine is absent. So now we are going to discuss about like a summary table, okay, to summarize all the formations of these ribonucleotides and deoxyribonucleotides, and we will see how to name these nucleotides. Okay, so let's start with the summary table for ribonucleotides, then we will discuss about deoxyribonucleotides. So firstly, as we know that we are having a nitrogenous base, and if we add a pentose sugar on the nitrogenous base, then it results in the formation of a nucleoside. If the nucleoside is added with a phosphate group, then it results in the formation of finally a nucleotide. So this whole process we know very well. Okay, so let's start to draw the table for ribonucleotides. There are 4 ribonucleotides. First one is like on the basis of nitrogenous bases. For the nitrogenous base, first one is adenine. If on the adenine we added a pentose sugar, the pentose sugar will be, yes, the ribose sugar. Then it results in the formation of adenosine. Learn the names of these nucleosides. Nucleoside is adenosine. adenosine. Okay, and now if this adenosine is further added by a phosphate group, or if I say if the phosphorylation takes place of the adenosine and one phosphate group is added, then it forms adenosine monophosphate. Okay, adenosine monophosphate can also be called as adenylic acid, adenylic acid or adenylate. Adenylate. So now second one is guanine. And we added ribose sugar on guanine. It results in the formation of guanosine, and if guanosine is again phosphorylated and we added a phosphate group on this, it results in the formation of GMP, which is also known as guanylic acid or guanylate. Okay, the third one is cytosine, and if we added a ribose sugar on cytosine, it results in the formation of cytidine. Cytidine, not guanosine, so cytonosine or something else, but it is cytidine. And then phosphorylation of cytidine takes place, and after phosphorylation the nucleotide which is formed is cytidine monophosphate or cytidylic acid. And the finally fourth one is uracil, and then the nucleoside is formed is uridine. And if further phosphorylation of uridine takes place, it results in the formation of uridine monophosphate, which is also termed as uridylic acid. Okay, so this was all about ribonucleotides. So without wasting our time, let's move towards deoxyribonucleotides. So now the first nitrogenous base is adenine. Now for deoxyribonucleotides, which pentose sugar are we gonna use is, yes, deoxyribose. Okay, if we added deoxyribose on adenine, then the nucleoside formed is deoxyadenosine. And if the phosphorylation takes place for deoxyadenosine, then the nucleotide formed is deoxyadenosine monophosphate, and this is also known as deoxyadenylic acid. Now for guanine, if we added deoxyribose sugar on guanine, the nucleoside formed is deoxyguanosine, and if phosphorylation of deoxyguanosine takes place, it results in the formation of deoxyguanosine monophosphate, which is also known as deoxyguanylic acid. Now cytosine, if we added deoxyribose sugar on cytosine, it results in the formation of deoxycytidine, and deoxycytidine is, if phosphorylated, then it results in the formation of deoxycytidine monophosphate or deoxycytidilic acid. And the fourth one comes is thymine. If we added a deoxyribose sugar on thymine, then it results in the formation of deoxythymidine. And if phosphorylation of deoxythymidine takes place, it results in the formation of deoxythymidine monophosphate, which is also known as thymidilic acid, thymidilic acid. Okay, so now we completed ribonucleotides and deoxyribonucleotides. In this summary table, the question which can be asked is just what type of sugar is present. Like the examiner will give you anything from this, like DAMP or anything. Okay, so he will ask you, he will give you some name from all these, or maybe the more phosphorylated form of these nucleotides. That ATP, in ATP what type of sugar is present. Okay, and the examiner will give you four options. One will be ribose sugar, deoxyribose sugar, both, or some other sugar name the examiner will give you. So we know in D, ATP, DMP or anything, the sugar present is deoxyribose, but in ATP, the ribonucleotides, the sugar is present is ribose sugar, not deoxyribose sugar. Okay, so yes, one more thing. The nucleotides which we have formed now for deoxyribonucleotides and for ribonucleotides are in inactivated form. These are of inactivated form of ribonucleotide, deoxyribonucleotides or nucleotides, we can say. These nucleotides, the monophosphate form of nucleotides are inactive. Okay, when there is like replication or something, transcription, etc., where we need nucleotides, then these inactivated nucleotides are activated by further phosphorylation of these. Okay, so like in RNA replication, these inactive nucleotides like AMP, GMP, CMP and UMP are further phosphorylated to form the triphosphate form, which is the active form, and then these nucleotides are further bonded each other to form the RNA or DNA. So now we have completed all about nucleotides and their basic structures, and there are some purine and pyrimidine and nucleotide analogs which are clinically used in treatment of many of the diseases. So the only thing now we are just gonna do firstly are looking after for that nucleotide analogs. Okay, so which are very important in clinical medicine to treat the many of the diseases. So here goes purine, pyrimidine and nucleotide analogs. Okay, so First one is allopurinol. Allopurinol, which is used for the treatment of gout or hyperuricemia. Okay. Hyperuricemia is a situation in which uric acid is excreted in excess, is formed in excess amount and get excreted through the urine. Okay, and gout is a joint related disease. Okay, and the second one, gout or hyperuricemia. So like gout is also a disease related to hyper excretion or hyperformation of uric acid. Okay, the second one is 5-fluorouracil. The third one is 6-mercaptopurine, and the fourth one is 8-azoguanine. 5-fluorouracil, 6-mercaptopurine, and 8-azoguanine. These all three are used in the treatment of cancer, like esophageal cancers, cervical cancer. Example. Okay. The fifth one comes here is azathioprine. The azathioprine, when gets inside our body, it is converted to 6-mercaptopurine, and then helps in treating the autoimmune diseases. Like arthritis. The sixth one comes is arabinosyl adenine, which is also like denoted as small a RA, arabinosyl adenine. Okay, this is used in the treatment of neurological diseases. For example, viral encephalitis. And the seventh one is arabinosyl cytosine, which is also represented as ARA capital C. It helps in the treatment of cancers like leukemias. Okay, so and the last one is zidovudine. Zidovudine. The zidovudine is used for the treatment of HIV or AIDS. So now the first part of our video is completed. I hope you all are like able to understand what I wanted to make you understand. So if you all like this video, so please if you have not subscribed my channel yet, please subscribe to my channel, and please, I'm looking forward for all of your suggestions, and your suggestions are very precious to me. And thank you so much.